This is Perry Marshall, and I am delighted to be here with Michael Levin. He is, well, I mentioned him to my friend Dennis Noble, and he said, oh, he's a brilliant electrophysiologist. I think that is the title that he gave to you. So I don't know if that's your official title, but that's what a physiologist had to say.
That's great. I love that, Dennis. I'm not an electrophysiologist, but I love Dennis, and we can certainly talk about electrophysiology.
And well, the way I got introduced to Michael was one of my very long-term clients and also volunteers on the Evolution 2.0 project, Paul Boscheman, who is a chemist. I believe he used to work for DuPont. He got a hold of your videos, and he's like, dude, like, this is like really good stuff. Perry, you gotta watch this. And so he sends me this video, and I'm a person who probably prefers to read rather than watch a video, and I'll put a link in the show notes. But I'm watching this presentation, and he's Michael's cutting planarian worms in half and getting them to grow two heads, and Michael is he's implanting tumors in creatures and watching how they work and changing electric fields and making the tumors go away. And he's finding out that you can put an eye on the spinal cord of a embryo, and the eye can actually see, and they don't even have to tell the thing how to rewire the nervous system. And it's just amazing things. And Michael, one of the things that really stood out was the number of times that you said things like, we have no idea how this works, but it happens. We have no idea how this works, but it happens. We have no model for this, but it happens. And my take was, okay, this is a guy who, scientifically speaking, dances on the wing of the airplane where most people are trying to be safe inside. And I love that because it's like, I occasionally run into articles and authors where they make it sound like we've got everything figured out. I don't think we've got 1% of it figured out.
Bingo.
Do we even have 0.1% of it figured out? Like, there are vast regions of dark matter in science that we just have no clue. And there's a humility in that. And that, Michael, that is what I liked the most about your work. And I watched this video twice. I watched it again last night. It's like, okay, I think I'm starting to get some of this. Most of it's way outside of my, familiarity, but okay, I get what you're saying. Wow. So I guess that's the intro. That's who. Yeah. And Tufts University near Boston. So Michael, let's just start with the kind of the elephant in the room. You're at a big university, and big universities have to send all the students home, and the world is in this crazy virus phase. So you've got to get out of bed every morning. You've still got to do research. You've still got to get your research teams. Just give me a snapshot of, like, how are you managing all of this, and what's going on?
Yeah. Well, I think first, what I'd like to just briefly address the thing you just said about the areas of ignorance and so on. I think this is really profound, and I want to say two quick things about that just right off the bat. When I point out in my talks that there are lots of things that we don't know how they work, the point is not that we seek to collect sort of weird phenomena and leave them at that, right? So that's clearly, we would like to understand how things work, and in fact, we've reached, I think, and hopefully we'll talk about that today, some really important new discoveries about how all this stuff happens. At the same time, I think it's very important to keep ourselves and our students honest as scientists about what we do and do not know. know. And in fact, identifying these areas of ignorance is really critical for progress in the future. I mean, just as you said, if you read some of these reviews or especially textbooks, you get this feeling that, hey, we're doing pretty well and there's just some stuff to clean up, but overall, we've got this under control. And I think that's profoundly wrong. I think you're much closer to this 1% figure as far as what do we actually understand. And I think it's really important, especially for people outside the field, because there's a lot of, I speak to a lot of different audiences, computer scientists, roboticists, and they have a feeling that biology has all these things explained, and they really don't often know where the deep questions are for even within biology, within different fields. People kind of have this assumption that stuff is well handled, and so I really like part of my job is to poke at these places where actually things are not nearly as clear as everyone thinks that they are. So I like that a lot. You use the phrase dark matter. It's quite interesting. When the late Paul Allen in 2016, when he created our Allen Discovery Center around our work at Tufts, he very explicitly used this phrase. He called it to explore the dark matter of biology. So he very specifically wanted to fund a center that was going to map out areas where we don't even know what all the right questions are yet, and he was very conscious about going after places where clearly there's something important there, but we don't even know what it is yet. And so I think we are looking at some really fascinating areas in the dark matter of biology. This is now, of course, more challenging now because of all this the quarantines and being shut out of our labs. At the moment, our situation is simply that all experiments are on hold. We do, of course, have ant colonies and flatworm colonies and frog habitats, and all of these things are being maintained by some of our intrepid people in the lab who still have to go in and make sure that the animals are doing well and so on. But the rest of us are at home, and we are basically, we coordinate all the time. I think I'm busier than I ever have been during these weeks as far as checking in with everyone. We are writing. My coders are coding. The computational people are making models and analyzing them. We are writing up manuscripts for tons of stuff that we have open. That was in progress basically when all this happened. We were sitting on, I don't know, probably 20 different manuscripts from the stuff that was going on when we all got told to go home. So for now, that's it. It's luckily a good chunk of my lab is computational and theoretical, and so there are people that can still do a lot of work on the in silico, so to speak, and everybody else is writing.
Well, so how are you received? Because usually people that are telling us what we don't know are kind of black sheep. Like, hey, we had this really wonderful party going on, and you had to come in here and fart in it. Like, do you get pushback? Like, how is politically and socially, how does it work for you?
Yeah, I've noticed a number of really interesting phenomena. One thing I'll say that I've noticed is that computer scientists and engineers tend to be really very, very open with this kind of stuff. So just as an example, a couple of years ago, I was at the Neuro IPS meeting in Montreal, and it's something like 6,000 machine learning folks, and pretty much everybody in the room is doing the variants of deep learning, and everybody's working on how to store memories in very precisely tuned synaptic connections of these deep networks, right? And so I gave a talk where the first 10 minutes of the talk was going over this example of, I went over two examples. The first is the butterfly example, where you take a caterpillar and you train the caterpillar for a particular memory, and then the caterpillar metamorphoses into a butterfly. And during this process, it has to completely rebuild its brain. The brain is basically taken apart. Many of the cells die. Most of the cells lose their connections, because you're going from a soft-bodied robot that has to crawl and chew plants to a hard-bodied creature that's going to fly and drink nectar. So the whole brain is just totally redone. And guess what? The butterfly still remembers the original information. Now, what is this telling you? If you're somebody who sunk, you're Google or Microsoft, who sunk all this money into deep learning, you're now finding out that, hey, there's a biological medium out there that where the memory remains even though all of the connections get torn to shreds. So it's not in the synapses. So where is it, right? And so, but what was amazing is that and then I talked about our planaria work, where heck, you can cut off the brain entirely, and then they regenerate a brand new brain, and the memories come back. And so that audience was incredibly welcoming. I mean, my phone blew up at that point. People were extremely interested in it because, and it was a risk. I didn't know how people would take it. I mean, as you said, that's a party that everybody's kind of very happy to be at. I didn't know how that would go, but people were fascinated, and I think it's because they're engineers. It's because they don't have as much preconception about how things are supposed to work as much as they're interested in how things could work better. So everybody was pretty excited about it. That is definitely not always how my reception goes. Sometimes people are really freaked out by some of this stuff. What's interesting is that I find, so I give this talk to lots of audiences, regenerative medicine folks, cancer biology, developmental genetics, all over the place. And I can always tell what kind of department I'm in based on which part of my talk makes people mad. And what's interesting is that it's always a different part. Things you can say in a molecular, in a neuroscience department that are extremely obvious. You say the same thing in a molecular genetics crowd, and people throw tomatoes. And it's not right because we have these amazing silos where people get used to thinking about things in a particular way, and then anything that goes against that becomes really disturbing to them. And again, there are things that you could say to a regenerative medicine crowd that's totally fine, but the same thing in a traditional cancer biology meeting is a big problem. And these kinds of things are all over the place. So I, at this point, am totally fine with that. I like a good mix of making people intellectually uncomfortable with exciting them about the possibilities. Yeah, so there are all sorts of aspects of my message that don't sit well with particular sort of paradigms, and then there are other aspects that people are very excited about because they point to a way of making progress in areas that are going to be otherwise very hard to deal with.
Well, Richard Fineman said something to the effect of, "That which I cannot build, I do not understand."
Yep.
And I think that the gold standard for scientific theories is engineering. And I need to explain what I do and don't mean by that. I don't mean that just because it works means your theory is right. But what I do mean is if it doesn't work, your theory is wrong. Right? And when I went down the rabbit hole 15 years ago in evolutionary biology as an electrical engineer, and I start reading evolution books and papers, and I'm going, most of what they're saying makes no sense. I could never build a computer software program with random mutation and natural selection all by itself. Right? I would need a whole bunch of other mechanisms. And then eventually I started finding these in biology. They just weren't being talked about in most of the popular books. Well, so let me tell you a little story. I for reasons I won't go into, because it's a whole nother story. I started doing interviews with scientists and asking them the question, on a scale of 1 to 10, 1 being good, 10 being bad, how bad is the politicization, polarization, difficulty of publishing, difficulty of getting grants, the bureaucracy, and political correctness? Like, how bad is it in your field? And when I talk to physicists and chemists, I got 3, 4 answers like that. When I talk to people in cancer and evolutionary biology, I get answers like 8. And one researcher who has done both of those, all of those fields actually, he said, I think I can tell you why. He says, theoretical physics is a 3, and that's because the foundations of theoretical physics are very well understood. Nobody's arguing too much about that. He said, we still don't know what the foundations of biology are. And we still don't know what the foundations of cancer are. And so these fields develop dogmas that people cling to with certainty. And then if you show up and you have something that's against the zeitgeist, well, good luck getting something through peer review. That's how he described it. Would you say that what you're experiencing, like your own version of that, where you go to these different departments and they're sacred cows and taboos, is, give me a reaction.
Yeah, I think that certainly exists, and it's definitely an issue in some for some things. I think two things need to be said. I want to be clear that I'm not complaining in the sense that, I mean, over my career we've been well funded. We publish all the time in mainstream journals, so I'm not complaining by any means. I'm not saying I've been unfairly treated by anybody. I don't have any axe to grind against the mainstream structures. I'm not complaining. Having said that, the reality is that yes, absolutely, we spend an inordinate amount of time writing and rewriting and massaging things in a way that is designed not to freak anybody out, as opposed to just sort of putting things in the way that I think are the most accurate in terms of what I think is going on. Obviously, this isn't for primary data. This is for what do we think it means, right? That kind of stuff. And I think we have to spend a lot of time thinking about how to pitch something to specific audiences. And then part of that is worthwhile. We want our, I certainly want my my more junior scientists to think about how things are going to be best absorbed so that they can actually make an impact and not write these things that nobody is ever gonna read. So part of that is worthwhile. But for sure there are problems, and I think that most of these problems arise not because anybody is evil or trying to suppress new knowledge, or there's, I don't really, I have not seen any evidence of any kind of major conspiracies going on. I think most of these things are kind of inescapable limitations of humanity, meaning, everybody's only got so much attention span because the amount of knowledge is doubling. I forget how fast, but very rapidly. You've only got so much stuff to pay attention to. And so everybody has to kind of make this cutoff. Okay, here's this thing that's really weird. Am I going to dig into it now, or am I just going to sort of wait till it shows up in the textbook where everybody else has vetted, and then I'll pay attention? So that's the sort of thing we face a lot more than, boy, I hate this stuff. I'm going to make sure nobody ever sees it. I've honestly, in my 25 years doing this now, I've not come across much of that intentional obfuscation. It's mostly just, how do I make this stuff so attractive to people that they have to fit it into their busy day? So, I think there is absolutely an inertia to science. I think there is absolutely a lack of imagination when you standardize. You have a study section or something like this, and you've sort of averaged everybody's unique gifts, and what do you end up with? You end up with kind of a middle ground that's kind of this muddy sort of middle that's not very good at discerning the really brilliant ideas from the kind of crazy stuff. So I think these are limitations that we all have to work to overcome, and I don't think most of it is anybody trying to go down the wrong path.
Oh, so when you go when you go to a particular group and you sense that you've knocked over one of these sacred cows, then you're not getting hostility. You're just getting, what is it? Bewilderment? Is it?
Well, it's everything, and I'm not going to say I haven't gotten hostility, because I certainly have, especially in things like anonymous peer review. I mean, boy, once you make something, and once you give somebody the cloak of anonymity, then they really go at it. But my view on this, and what I try to impart to the people in my lab, is that it's not personal. I don't think anybody really cares about me in the sense of, boy, we're gonna make sure that this guy his stuff doesn't come to light. I don't think anybody's thinking about me in that way. It's not personal. It's here's somebody who's thought about things in a particular way. can I do a good enough job of getting my point across so that they are motivated to think about things in a different way? Now, that may be impossible for some people, but I found that largely it's quite possible. And this is also why, so I have a very limited travel schedule for work and family reasons. When I'm invited by graduate student associations, I always go. Because if you think about it, when you if I give a talk to a large audience of well-funded PIs who have, they're funded for the next 10 years to do exactly what they're doing, how much impact am I really going to have? Who's going to change, right? Who's going to change what they do based on anything that I tell them? Whereas the postdocs and the grad students in the audience, their brains are still kind of soft. They're still thinking about what are they going to do in the future? What are the big questions? How do they want to think about things? And that's where I think all the impact happens. I frankly think that you're much more likely to impact science going forward by presenting another option to the young people who haven't really crystallized yet. So again, I never take this stuff personally, although certainly this stuff happens and sometimes there is hostility. But I think it's on me, and I tell my people it's on us to present this in a way that it's inescapably compelling. If somebody's just not getting it, part of it's on them, but part of it's on you, because you have to make your stuff so clear. And I think I think that's probably why in physics there's less of it, because in physics and math, I think you have a greater opportunity to make something that's just inescapable. Here's what it is, and you follow and you see that I haven't made a mistake between lines 1 and 10. The conclusion is what it is, and what are you going to say after that? Biology isn't really like that. There's a lot of room for interpretation. But I really think it's on us to make sure that our interpretation of things is so compelling that people have no choice but to listen. And to a large extent, I'm happy with how that's gone. I don't really have any complaints about it.
Well, that makes a lot of sense. And it also makes sense to me because I make my living in marketing. And, I think most of what most people think of as science is really marketing. And you use the word pitch, and that is the right word. How do I, a scientific paper, like it or not, is a sales pitch for a particular way of doing things. And the question is, did you prove that what you're pitching is correct? And I think that's also true in marketing. If it's an infomercial with an exercise machine, did you prove that the exercise machine is really going to make you healthier, or are you just going to shove it under your bed and forget about it, right? So
that's exactly right. That's exactly right. And people, people complain a lot about the editors at various journals and how you have to appeal to them. Okay, but the real battle is appealing to the limited time of the community. What you really want is you want eyeballs on your stuff, and you want people who are in the position to do something with it and to move the ball forward. You're competing. It's very much that aspect of marketing, not because it's got to be fluff and sort of dressed up in a way that it's not. I don't mean that. I mean you literally are competing for the limited attention. We all have only 24 hours in our day, right? And the people who are the most effective at getting stuff done, their time is the most limited. So how are you going to get them to read and understand what you're talking about? That's really the battle.
Yeah. Well, this is great. So let's shift gears a little bit. Let's talk about, there was a very interesting part in your video, which again, I'll put in the show notes, where you said there are two views of cancer and regeneration. So everybody knows what cancer is, but what regeneration is if I cut the leg off of the frog, does it grow back? If the kid smashes his end of his finger in a car door, does the finger grow back? And with young children, they actually do. And you've studied this in great depth. And you said in your video that there are two schools of thought about how regeneration relates to cancer. Could you explain the two views and what you've been doing with this?
Sure. The first thing I want to do is enlarge a little bit what we mean by regeneration. So broadly speaking, the point of regeneration is not simply to grow back whatever was lost, but think about the problem that these cells have when something is lost. So let's say you're a salamander, and let's say somebody cuts off your arm. One of the things that the cells have to do is that they have to be able to they start growing, they start patterning, they're making this limb. The biggest thing for them to do is two things. Number one, to know what to do to make a limb, okay? And number two, to stop when they're done. And hardly anybody studies this last point because it's super critical. These cells are being called upon collectively to make a decision about a massively large measurable, the size and shape of an arm. And in fact, in some cases, this is actually remodeling, and I'll talk about some frog data where it's not, you're not regrowing it from scratch, you're actually remodeling things in place, you're moving stuff around. So so it's a massive computational problem for them to know, hey, what does a standard salamander arm look like? And have we reached it? And can we stop? Now, to an engineer, or a cyberneticist, this is pretty obvious. It's error minimization. So you've got a set point. You know what your target looks like. You take a step, and you keep taking those steps. Kind of a means-ends analysis. You keep taking those steps until the error is zero or as small as you're willing to live with. Okay, that all presupposes that you know what the target is that you're trying to reach, and we'll talk about that in a minute as well. But the whole point of regeneration is not just regrowing stuff back, it's the ability to exert control over very small things, which are your cells and the pathways inside those cells, to achieve a very large goal state. So for me, the important thing about regeneration is that it's very much a pattern homeostatic process. It is the ability of the system collectively to harness the activity of cells towards a very large scale outcome in anatomy, an anatomical state. So we see this in regulative development where, let's say, you can take mammalian embryos, cut them in half, and you still get two perfectly normal monozygotic twins. Or in regeneration, these are all instances of the same thing. They're the ability of cells to work together towards a global goal. Okay, so now we can talk about how this relates to cancer. So there are two basic views on cancer. Speaking very kind of roughly, the more popular modern view, although I think the cracks are now pretty deep in it, but the more modern view is that what happens in cancer is that you basically have a series of mutations that have irrevocably damaged a cell to the point where the typical growth controls are off. So now this thing is going to grow out of control. It will have clonal progeny, which will make a tumor. They'll metastasize, blah, blah, blah. So that view makes a very specific prediction. If you believe that that's what cancer is, then what it suggests is that animals which have very ready access to large numbers of proliferative cells, of plastic cells that don't know what they should be yet, should be very cancer prone because because this view is at the cellular level. It's a view that's focused on checkpoints from yeast and cell cycle control proteins and things like this that focus on the individual cell and talk about what happens when you have cells that are actually pretty comfortable proliferating like crazy, like you would have to have in a regenerative, in a highly regenerative animal to get a whole limb back. I mean, the rate of proliferation is incredible. It's faster than any tumor. And so, so the prediction would be that animals that are good regenerators would have huge cancer loads because you have all these cells around that are really ready to proliferate, and they're plastic. They can be different types of cells, and so. So that would be the prediction that you would make. That prediction is incorrect. So it turns out that animals that are good regenerators have in fact the lowest rates of cancer, and to the point where there's even older data where you can induce tumors on, let's say, the limb of a salamander, cut through the middle of the tumor, and the regenerative process, and this has been done in salamanders, it's been done in planaria, it's been done in more recently in the 80s and 90s by Mary Hendricks in chicken and fish embryos. There are these highly regenerative environments actually normalize cancer. They normalize the behavior, and you think, why would that be?
What do you mean normalize?
What I mean normalize is that they become, and we've done this in our lab electrically as well, that we can talk about. They instead of killing the cells, they convert them to normal behavior where they participate in morphogenesis with the rest of the tissue.
Oh.
This was originally found a long time ago. Beatrice Mintz showed this in the late 70s. You can inject human carcinoma cells into a mouse embryo and they become part of a normal mouse. So why would that be? So the second view of cancer is completely different. The second view of cancer is more top down, whereas the first one is bottom up. And the top down view says, look, what you have, the real question isn't why is there cancer? The question is, why is there ever anything but cancer? I mean, we have this profound question of how do you take very competent individual cells, which are normally, they're amoebas, they're crawling around doing interesting things. You now have to convince them to work together to build and repair a giant thing. Their goal is now huge, as opposed to the boundary that they normally care about, which is just the boundary of a single cell. So normally their self, okay, and so I'm going to use this weird concept that kind of drives a lot of people crazy. But if you think about what is the scale of the self of a single cell, it's basically the boundary of the cell, and all of its goals are constrained within that boundary. All it cares about is dividing, proliferating, going somewhere where life is good, and this is metastasis, whatever. The question is, how do you get those cells to work together towards a much larger goal, like make a body or make a limb or an organ? So the other view is that animals that have a very good, powerful control structure that is able to harness cells towards these large-scale purposes are cancer resistant, because when a single cell is sort of teetering on this possibility of, hey, I'm going to contract my scale of self to just handle my local problem, and I'm going to crawl around and treat the rest of the body as just environment, they have powerful cues that are constantly operating in morphostasis. They're constantly operating to keep everybody working towards a bigger goal. It's a motivational issue, so to speak. It's a way to keep all of the cells engaged in this process of anatomical homeostasis, where what they're trying to achieve is not a single cell goal, like make as many copies of myself as I can, but they're actually working on a much larger goal, a morphogenetic goal of making a limb or whatever. And we can talk about, I mean, it sounds very sort of anthropomorphic, but there's actually some very obvious, rigorous biophysical models of how this works. So this other idea is that, yeah, regenerative environments normalize cancer because they prevent cells from going off on their own, and they keep them harnessed towards this large-scale morphogenetic plan. And so this is this is much more consistent with the data. And in fact, this is why as far back as the 80s, people knew that one of the first steps towards tumorigenic transformation is the loss of what's called gap junctional connectivity. So what I mean by that is there are these little electrical synapses. They're sort of an ancient version of our modern neural synapses that allow two cells to directly share with each other their electrical and physiological state. You can think of them as almost submarine hatches that sort of dock together like this. They allow two cells to come together and to dock so that small molecules from one cell can go directly in the other. And they're very clever. They can open and close and so on. The first thing that happens when these things are shut off is that the cell is now no longer perceiving the electrical signals from the network. The cell is now an individual, and as far as it's concerned, its computational boundary has shrunk to the point where it no longer feels all of the instructions that are happening around it from these other cells, but it begins to treat them as an environment. The boundary between self and world shrinks, right? And so now that cell can go crazy and proliferate and do all these different things. And so what we've shown, and now other people have shown, is that you can actually, this makes some very specific predictions that we've tested. So for example, when you take cells, genetically perfectly normal cells, no carcinogens, no oncogenes, nothing like that, perfectly normal animals, and you artificially manipulate, in fact, block this normal electrical coordination between the cells, you trigger cancer. And we can make a metastatic melanoma in perfectly wild type, let's say Xenopus tadpoles, purely by deranging the ability of these cells to talk to each other electrically. There's nothing genetically wrong with them. Even better, what you can do is you can take human oncogenes, throw them into these tadpoles, which normally cause tumors. If you do that, but at the same time you force the appropriate electrical communication between the cells, you keep them plugged into this large network that makes decisions about, hey, what are we making? We're not making a tumor, we're making an arm or a kidney or whatever. If you force this electrical connection, then despite the really strong presence of the oncogene, and we're talking KRAS and things like this, you will not get a tumor, and you will get normal cells that participate in normal morphogenetic functions. Now, I'm not saying that this cures every possible kind of cancer. I'm saying cancer is not one thing. There are many different phenomena here. But there's a really important sense in which cancer is not a single cell disease. It is a disease of communication. It's a disease of boundary between self and world, and not letting that boundary shrink to the level of a single cell, but keeping it at the network level, keeping it at the tissue or organ level.
So cancer is a distortion of the cellular sense of self.
I think that's very true. Now, I'm not going to claim that there's only one correct view of cancer. Okay, so there are other people, right? There's other people that have useful paradigms involving, game theory and various other ways of looking at cancer. I'm sure some of those are useful lenses through which to see this problem. Yeah. What we work on now is exactly this boundary of the self. And so I see evolutionarily, I see the rise of multicellularity as the expansion of the self from a single cell level to a large collective that has goals befitting a large collective, so scaled in space and time. So morphogenesis, pattern memory, and anticipation, and so on. And then cancer is a perhaps temporary reduction of that inflation back down. And I think this sense of self is it's dynamic, so it can grow and shrink during the lifetime of an individual at any level, so cells, tissues, and so on. And managing this is an important strategy going forward for biomedicine.
So you sent me a couple months ago a bunch of papers you'd written, and one of them was in some kind of a psychology journal, and it was about biological sense of self. It was something along that line.
Frontiers in psychology or something like that.
Yeah. And you, in that paper, you talked about how you are willingly anthropomorphizing biological behavior. You're comparing it to humans, and you pointed out that there's also the excessive prohibition of anthropomorphisms, and traditionally in molecular biology, these kinds of anthropomorphisms have been very, very frowned upon. And my sense is that you can't make sense of this unless you invoke these kind of analogies. And in fact, I can't help but think of, even viruses, hot topic lately, are also changing the identity of cells too. That maybe you could even frame the virus problem in a similar light. Do you have any comments to make about that?
So I don't have any wisdom on viruses specifically, but I want to talk about this anthropomorphic thing. It's a very interesting phenomenon. So as of course you're right, molecular biologists hate that kind of stuff. And what's interesting about it is that on the one hand, at least in theory, what they're trying to do is avoid any kind of non-rigorous, sort of woolly thinking. But in reality, by calling it anthropomorphic, so the root there is human, right? That's what you're going for. You're really breaking with the modern understanding of evolution, because these things that we're talking about, so memory, goal-directedness, learning, perception, all of these things, they didn't just show up in humans. I mean, it's completely magical thinking. I'm constantly, just amazed at how people will say, "You're telling me that this tissue can have memory?" No, we have memory. I say, what do you think you are, magic? I mean, you don't have anything that hasn't been around evolutionarily for probably 1 billion years, and so. We need to now take very seriously the fact that all of the things that we think about as higher animal kinds of cognition, they have an evolutionary history. They don't just show up suddenly because we're humans and we're cool. This stuff has been around forever. And all of the molecular biology is catching up now, because now we know that our unicellular ancestors had all of the same, just from all the genomics we now know, our unicellular ancestors had all of the synaptic machinery we associate now with complex neurons. All of this stuff, you think biology wasn't doing learning and anticipation before until humans came along? I mean, it's crazy. It's magical thinking. And I always, when I talk to people about this, and they say, how dare you, use these words like memory and learning for tissue. I mean, we work on skin learning and things like this, and they say, oh, that's crazy talk. And I say, well, show me the magic that you think applies this to humans only. So I think the most important thing is for everybody to take evolution very seriously and realize that all of the cognitive types of capacities that we talk about have a phylogenetic history. They were here in simple forms. Obviously, I'm not telling you that bacteria are quietly writing essays when you're not watching. What I'm saying is that all of the things that we associate with higher cognition have basal analogs that have been slowly ramped up during evolution, but they've been here since the time of bacterial biofilms. So this kind of essentialist binary thinking that there's real cognition, which is what humans, maybe the great apes, do, and then there's everything else, which people talk about that as cognition, but that's not really that should have been gone from shortly after Darwin. Like that is no longer, that's a view that's no longer tenable. It's not, you can't maintain that seriously anymore. And so it's completely reasonable to use computational models of things like decision making, memory, anticipation, self models, world models, all of that stuff, to use it to try to explain decision making during, for example, regeneration. How does a regenerating collective make decisions? So people say, well, only brains make decisions. Well, for example, we had a meeting a while back, a basal cognition meeting, and somebody wanted to talk about the origin of neurons. So he kind of got up and he wrote down four characteristics of neurons, and I immediately said, well, every cell in the body does this. And so then we had a 2-hour discussion of what exactly is a neuron. And guess what we found out is that there are some specializations, like the special shape and the speed with which it acts, but all of the things neural networks do, cell networks have been doing long before neurons and brains showed up. Bacterial biofilms do this. There's some very nice recent work on that. So my point, I often start these talks with a continuum slide with a bunch of different types of cognitive properties on it, starting with the very simple things that even bacteria can do, and going all the way up. And so I think that there's no such thing as really anthropomorphizing. There's only one, and this is a good point. It gets back to your point about the engineering. I don't think that any of these things can be handled at the level of philosophy, where somebody says, I fundamentally don't believe in using anthropomorphic concepts below the higher mammals, and somebody else says, well, I do. Okay, that's great, and that's all philosophy. It gets us nowhere. What I think we need is a very clear engineering approach. The thing is very simple. Here's a model of memory and learning that I'm going to apply to a bunch of skin cells. Now, all we're going to do is we're going to find out, does that model get us to better experiments and better control in a regenerative medicine setting, or doesn't it? If after some number of years of working with this, we've gotten nowhere, then I will gladly say, okay, guess what? This was an inappropriate metaphor. It's gotten us nowhere, and that's that. On the other hand, if it gets us to better understanding and control of what's going on, then you have to say, no, it was a good metaphor. It was exactly appropriate, and it helped us get to new science and new capabilities. So I take a very pragmatic approach about it's sort of akin to Dennett's intentional stance, where he says, it makes sense to treat an agent as cognitive when that gives you the most ability to understand what the thing is about to do. And that's exactly what we're after. And I think regenerative medicine folks take to this pretty well, because in the end, they want an outcome. And if I tell them that we can get better regeneration by motivating a cellular collective with rewards and punishments, they say, fine, do whatever you got to do. Let's get that finger growing back. Whereas people who really are committed to a bottom-up perspective are against this kind of idea on fundamental grounds, which to me is nowhere. We need to be engineers about these things.
So if I take your engineering modeling approach and I said, well, I have a hypothesis that not only does cognition happen in cells, but bacteria are better programmers than humans, and maybe my stomach has better memory than my brain, because I just had a senior moment yesterday because I couldn't think of somebody's name. Like, would your research actually support that at the lowest levels of biology, there's actually more intelligence than there is, you know, in the, you know, in a room full of PhDs.
Well, it's a hard thing because intelligence. Well, a couple of things. I mean, intelligence. I think, and certainly I don't have a unique definition of it that hasn't been said by other people before. But most likely, intelligence is not a scalar, right? It's not a single number. So when we say more intelligence or less intelligence, you know, anybody that's owned a parrot and a dog can tell you that they're both highly intelligent in completely different ways. So trying to say which one is more intelligent than the other is a tough task. I mean, if if you're thinking about intelligence as efficient problem solving, adaptability to novel environments, transference of lessons learned to new cases. I mean, bacterial populations, maybe not individual bacteria, but bacterial populations are certainly pretty dang good at it. And there are lots of people in the basal cognition field that are studying this. You know, I think certainly higher, so-called higher animals, including humans, have some unique cognitive capacities that don't exist at these lower levels. But I think comparing them is not trivial, you know, and I think we have to be ready to understand how to motivate your system by taking seriously the fact that it might be a cognitive agent. I mean, this is another, you know, this is something I for undergrads, when I first kind of introduced this whole idea, I say, look, you got a rat and you want this rat to do a circus trick. It's going to put a little ball in the little hoop, right? You got two fundamental approaches. You can try to control every single neuron in the rat's brain and play the thing like a puppet and try to get it to do what you want it to do. And someday, maybe that'll be possible. It's absolutely not possible now, but someday maybe that's possible. You can do something else, which is you can train the rat. And the reason that that works perfectly well is because you've now offloaded that whole complexity onto the animal. You're not worried about how it's going to do it. It's a top-down approach. You are providing a positive and negative reinforcement so that the system itself figures out how to do it. And guess what? It'll do the trick. And if you're trying to control it neuron by neuron, the sun will burn out before you get anywhere with that thing. So the reason that right? I mean, the reason that it's so much more effective, why is it so much more effective? It's because you have taken seriously the fact that your subject is a cognitive system that has goals. Its goal is to get more treats and less punishment, and it will do what it needs to do to get around. So some systems are very amenable to this kind of thing. The rat, obviously, if you want, you know, if you're dealing with a cuckoo clock, that's a terrible strategy because that system is not amenable to that strategy. So our goal as scientists, not philosophers, but scientists, is to not make blanket declarations about what is and is not appropriate, but actually to ask the simple question: what kind of strategy is best for your system? And then we find out. And so this is a completely an empirical question. It is not anything to sort of philosophize about, but people do have very strong preconceptions about what they think is right or wrong. But I think it only gets resolved at the bench.
So last question and we'll wrap up. I talked to you a month or two ago and I asked you a question. I said, what would you think of somebody who said, I think we need to make significant revisions to our evolutionary theories in order to really make sense of what all we know right now? And I could hear the emotion in your voice change, and I couldn't tell if you were not liking what I was saying or liking, but then in about 30 seconds, I could tell that you agreed. So could you just mention a few things that you feel like we need to leave behind and some things that we need to embrace from an evolution perspective in order to reckon with the things that are in front of us right now?
Yeah, right. So I could say a couple things. I guess the first thing that I want to say is to be super clear that some of the changes that people have proposed to this whole thing that come from a sincere wish that there was somebody orchestrating the whole thing from the beyond. This is not what I have in mind. Okay, so I'm absolutely committed to a naturalistic understanding of biology and physics, and I'm not saying that we got to throw this whole thing out and go with some sort of design hypothesis. That is absolutely not what I'm going for. At the same time, I think we absolutely have to acknowledge some important some important aspects of this that have currently been completely neglected. So a lot of what goes on addresses in developmental evolutionary biology addresses the hardware of the body, in the sense that people are very interested in the evolution of the proteins, the control structure of gene expression, and all of those things. Those are absolutely critical. All that work is great. It stands. We need it. What's still missing, however, is the true understanding of the software that hardware enables. There is amazing plasticity in that hardware. And I actually think regeneration is not about repairing yourself after injury. I think regeneration is for evolvability. And what I mean by that, so the damage that regeneration is initially meant to fix is mutagenic damage, not traumatic injury. So think about the following example. Two examples I'll just throw out there. One is that's been known for a long time, which is that if you, for example, have a developing vertebrate limb and you produce an extra bone, you don't just get a loose bone sitting there. In fact, you get an extra finger. So the other components they know what to do when they see bone, the tendons, the neurons, the blood vessels, right? So what this means is that because the system, and it's not regeneration in the classic sense, but it is in the sense that I mentioned, which is pattern homeostasis. All of the pieces are trying to make order at a particular level, at the level of a finger, out of whatever is going on. So this means that if there's some error or a mutation or something else that is going to screw up the system, all the stuff around it helps out. It helps to try to still make it a coherent organism. This means that it greatly flattens the evolutionary landscape, the fitness landscape, because now, I mean, one of the biggest problems with evolution, when you first tell somebody this who doesn't know about it, the theory of evolution, the first thing they say is, wait a minute, random changes in a complex system always make things worse. They hardly ever make things better. How is this whole thing supposed to work, right? That's the typical thing that everybody gets caught up on. So my point is that because the individual pieces are goal-directed, meaning not in a magical way, in a cybernetic way, meaning that it's, again, shaped by evolution, all the pieces are trying to reduce error from a specific homeostatic loop. These mutations that otherwise would have been deleterious and would have screwed up the system are now tolerable because everything around it still helps out. I'll give you another simple example. We discovered, so tadpoles, when they become frogs, they have to rearrange their face during metamorphosis. They have to move their eyes forward and their jaws come out. All this stuff has to happen. So it used to be thought that this was encoded somehow by the genome in a rote hardwired way. So if all tadpoles start out at a particular configuration, all frogs looks the same, well, then you just move all the different organs a particular way and then you get your frog. So what we did was we made Picasso tadpoles. We made tadpoles where everything is in the wrong place. So the eyes are back here, the mouth is over here, everything's messed up. Well, guess what? When you do that, all of the different organs move around in really weird and unnatural paths until they get to a pretty normal frog face, and then they stop. So what you find out is that what evolution has done here is not created a hardwired system that just does a particular thing. And there may be animals that are like that. Maybe C. elegans is like that. I don't know, the nematode. But what it's done here is produce a system that's much better than that. It's able to dynamically try to reduce the error between whatever's going on now and the target morphology that this thing has been evolutionarily selected for, to have a proper frog face. So I think that capability is what makes evolution go, broadly speaking. It's what enables all of these mutations to be at worst neutral and at best helpful, when otherwise they would have been deleterious. I think it greatly smooths out the evolutionary landscape because these things are trying to achieve specific ends. And so this is I mean, talk about things that get people all crazy. Talking about any kind of goal-directedness, it really drives people nuts. But I think it's very important to point out that while biologists are still sort of terrified of teleology and animism and all these kinds of things, engineers got over this in the 40s. So we've had control theory and cybernetics since the 40s and 50s, and everybody on that side of the aisle is super comfortable talking about non-magical artifacts that have that have goals and can pursue them. And this is like robotics and cybernetics has been all over this, and biology, that it really needs to catch up from that perspective, is to get over the fear of goal directedness, as long as it's cashed out by specific experimental work. So it's not mystery, it's not magic, it's not any kind of ghost from above trying to say what the goal is. It is a goal directed mechanism in the cybernetic sense. These are not magic. We have them in our house. They're the thermostats and such. It's time to expand the notion of homeostasis from single variables like temperature, pH, whatnot, to very large scale things like shape and anatomy. And I think this is a critical part that needs to be added to the modern synthesis. We've done some work on it, showing actually how to rewrite some of these goal states. That's actually, I think, some of the most important work from our lab, because making the hypothesis that these goal states exist, you ought to be able to rewrite them. If they exist, you should be able to go change the goal. And we've done that. And the cells then don't know any better, and they build to the new pattern. And this is some of our work on persistently two-headed planaria and things like this. So I think all of the existing work on evolution is super valuable. It's great, and it needs to be added to this more cybernetic perspective, where there is a multi-scale goal directedness. I mean, there have been now some nice papers on molecular networks that learn and have memory and have homeostasis. Obviously, cells do. Now we've been working on tissues and organs. This pervades biology, this multi-scale, and I've tried to push this idea to our roboticist colleagues, and they love it. People are making robots now based on this principle. But we in biology we already have such robots, and it's us, and we need to really add that sort of paradigm to the standard understanding of things.
Agreed. Michael, this has been fabulous. If somebody's geeked out by what they're reading, what website should they visit, or what should they go look at? That you would like them to start.
There are a couple of websites of mine that have everything and more that you might ever want to look at about these things. One is drmichaellevin.org, one word, drmichaellevin.org, and the other one is our Allen Center website, which is allenscenter.tufts.edu. And hopefully we can put links to them on your show notes.
We will do that. Michael, this has been fabulous. I love your willingness to run into these dark matter areas, and I'm expecting to meet you in person later in the fall. And
awesome.
And thank you very much.
Thank you very much. Yeah, great discussion. Thank you so much. Be well.